הכל חוץ מזה, אתה מבין?
בהסתברות, כן.
זה בדיוק.
פשוט יותר קל לבדוק את זה ככה. כי אתה פשוט רואה... מה נשמע?
חיים.
חיים? וואו. ממש... רחמנות.
זה החלק הקשה, שכזה הכל מתנגד כזה, והכל נהיה יותר קשה, ויש מבחן.
זה לא מבחן היחיד שלכם.
לא, אבל זה המבחן היחיד שהוא בתוך הסמסטר.
כן. טוב, זה מבחן בשביל להקל עליכם, לא בשביל...
זה מקל מצד אחד, אבל זה... מה זה?
זה צריך להיות מתישהו המבחן, כן. זה שאתה...
לא, בסדר, אין תלונות.
לא, אני גם מודע לזה שסטודנטים, זה מה שאתם יודעים לעשות. זה בסדר. להתלונן זה טוב.
להתלונן.
מה שאתם טרנד פה. טוב. Okay. So, this is, I know that you are heartbroken, but this is our last meeting, like a last lesson. Don't look at this, okay? And first of all, I want to say that there is, I think they already published the evaluation, like the online evaluation of the courses and feedback. So this is very important, especially in a class where there are not a lot of students. Then they only regard the statistics after there are more than 6 people that responded to the survey. So if you don't respond, then we'll not get any feedback, and it's very important. Even if you have bad things to say, then it's very important. Also because this is the first year that I'm giving this course, but you can also write good stuff. You know, don't have to write only bad stuff.
I apologize for deleting the email.
What?
I already deleted the email. I apologize.
You deleted the email of the survey? Yeah, you can. You'll see it. They will send it to you like 1,000 times. Don't worry. Okay. Yeah, they send each day or something like that. Okay, it's very. No, but it's important. Okay, so we'll start with going over the questions of the last chapter that we finished, and then we're going to talk about the article that I gave you to read, and then so hopefully we'll go over these pretty fast, and we'll end with a short. I hope it will be a short chapter about genetic techniques for like a how do you say, like a teaser for next course with Ami and Inbal. Okay, so which of the following determines if an mRNA fragment is considered to be an intron? Who wants to? And I will say that whoever answers the question cannot answer another question. Okay, so you can only answer one time. Okay, so someone who wants to try?
I can try, but I don't think I'm right.
Okay.
So there are specific electronic sequences, so that's right.
Okay.
I don't think the degradation rate of the removed intron is relevant
Okay.
for me. The specific exonic sequences, I'm not sure, like, why would that be relevant? Like, it could be relevant.
What about D?
As a thought. They track the exons, don't they? So, you could say that the areas of the mRNA that are not exons are introns, and that would make like C and D relevant, because C is like a specific areas to which our proteins attach.
Okay,
So
so
I guess A, C, and D, but I was
okay, so you're right, and actually, the degradation rate of the removed intron, a lot of times, so you need to discriminate between things. I will try also in the test to discriminate between things that we learned and were also in the presentation themselves. And I will normally not give you like questions. If you didn't see it in the presentation and you think that maybe it can be possible, then most of the time it will be like the wrong answer. Okay, so we didn't talk about the degradation rate of the removed introns. So although there is a constellation where this may have some kind of effect, but we didn't discuss that at all in respect to this. But we did say that there are SR proteins, and these are SR proteins bind to specific exonic sequences. Yeah.
I understood correctly that you look at an intern and if it's an intern or not.
Yeah.
I thought that the SR protein is the wrong answer because they affect to exons and not to introns.
So what?
you don't see them, more likely.
But are they?
Yeah, but what we defined in the previous class in the presentation that was talking about that, and that's why I say that you need to. Where is this? Just a minute. I'll just go to the. Normally, the question will be regarding like a specific thing that was in the presentation, and here, where is it? Okay. So, the definition of something like the exon recognition also is part of the identification of what is an intron, because it's not an exon, so it's an intron. Okay, so the association between these elements, these SR proteins, and and also these exonal elements or recognition sequences, are all this complex is part of the identification of an exon. But if you identify an exon, then it also defines the intron. Okay. Yeah, maybe, but it's directed towards exactly this slide. Okay, that you need to like understand that it's not that the definition. Obviously, a definition of an intron or an exon, they come together. Okay, if it's not an exon, then it's normally an intron. Okay. So.
And the specific exonic sequences are really the ones that SR protein is connected?
Yeah, they don't have to be like the same. Like you can have many types of SR proteins, but in general, these SR proteins know how to identify specific sequences inside the exon. Okay.
So that would be
Yeah. So the correct answer here is A, C, and D. Okay. So it's A, C, and D. Okay, easier questions now. Following is a list of processes that an mRNA undergoes during and after its transcription. Write the correct order of occurrence of these processes. So now you can't answer. Someone else? Yeah. Yeah. Right. Okay. Is it clear to everybody? Why? So, for example, this question was also in an exam in the previous years, and over there was also one of the options was polyadenylation, which this can be a little bit tricky because we talked about that in some instances the splicing or the recognition of the splicing element starts before the polyadenylation, but in general, what is classically known is that you have first the cap, then polyadenylation, then splicing, then export, and then translation. But again, I removed it so it won't be like confusing. Okay, next question. Who wants to try? A single-stranded? Okay. Right. 3 leads to cutting of the target mRNA. What about siRNA? It doesn't lead to cutting of the target mRNA? Okay. And 4? This is siRNA or microRNA? Okay. Okay. So almost correct, but 3 is actually relevant to both of them under some conditions. It depends on the protein that binds the mRNA. We'll see it also in the article when they discuss about AGO1 and AGO2 normally. And in the article they talk about AGO1. AGO1 has a more silencing effect if it's bound to a microRNA, like the protein. And AGO2, if it's bound to a microRNA, has a more like silencing and cleavage of the target mRNA. siRNA always leads to cleavage of the target. Okay. In general, and siRNA also has full complementary. You need to like this small interference RNA. You need to remember that the microRNA is like something that is normally from inside the body, and it's also they're highly conserved. Many times, much of the microRNAs are highly conserved, like small genes, and the siRNA are normally introduced by the experiment. Okay, so the experimental people who are doing the experiment normally they can inject the siRNA in order for it to function. Thus, a lot of time it has a full complementary of the gene because it's experimentally introduced. You want to have something specific. You don't want it to have like partial complementary, which introduces which enables this microRNA to target a large number of molecules, or different molecules. Okay, so remember what we talked about the microRNA, that it has a specific seed sequence, like 7 to 8 nucleotides only, so it can identify multiple types of mRNA, as opposed to the siRNA that has a full complementary sequence, and it can be like 21, 22 base pair long, which is enough for specificity. Okay, yeah. Yeah, but in general, they're not reported so much in animals. They're mostly found in plants. There are also but it's not like there can be in C. elegans or other organisms and stuff like that. But in general, we refer to them mostly like they're not coming from the actual organism, at least not the organism that we study.
And the second question, can you repeat what's the difference between AGO1 and AGO2?
Yeah, so we'll talk about it in the article more, but AGO1 has a more silencing effect. Doesn't have the endonuclease activity, which can cleave an mRNA in the middle. And AGO2 has also endonuclease activity. Okay, so the protein that binds the microRNA can determine also the functionality of the microRNA. Okay, so both of them can lead to cutting of the target mRNA. The molecule leads to temporal suppression that's only in microRNA. Undergoes perfect complementary, that's siRNA, and the molecule undergoes partial complementary. This is microRNA. Okay. So I wrote it here. What will be the outcome of cytoplasmatic polyadenylation? And again, just what was taught. Okay, not yeah, Jonathan. Daniel.
So it gets more stable. So like the opposite of A.
Okay.
It's one of the options down. Okay. Then recruitment of this protein transferred imaginary, like not directly but indirectly, yes. I mean, some proteins use the poly A to bind, and then who recruit other proteins to bind.
Yeah.
So the fixation of the mRNA back to the nucleus, back to the nucleus, like that doesn't sound right.
Okay. And
Increased gene transcription, yes, there's no poly A, then the gene is degraded, so there's no.
but transcription.
Transcription, no, translation.
Okay.
So
So out of these, what is the correct?
So B sounds recruitment of protein translation sounds correct.
Yeah. So in general, it's B. It's like you said, it's the opposite of A. It stabilizes the RNA if it has a longer poly A tail. Localization of the mRNA back to the nucleus. This is tricky because one of the things that we didn't go over, which I'm a little bit sorry about, but for next year is about the localization of proteins that certain proteins have different signals, like amino acid signals that can localize them to different compartments in the cell. Like a nuclear localization signal that is widely used a lot of time in proteins. If you want it to be present in high concentration inside the nucleus, you can put a small nuclear localization signal, like a sequence of amino acids that certain proteins in the cell know how to now to take this protein and ship it toward the nucleus. But we didn't learn about that, so this was like a tricky option if you learn about nuclear localization signals. And increased gene transcription, not directly. Okay, you can not. It can be that a certain mRNA that is a transcription factor will be translated, and then it will increase transcription. But in general, it doesn't. It's not a direct outcome.
Sort of like the opposite way around. Like, doesn't the increase in gene transcription cause more polyadenylation, but not more polyadenylation causes increase in gene?
No, it doesn't cause more polyadenylation. When the gene is made, a lot of time.
But it does cause like more like staples or something like that, which causes less field stimulation.
I'm not sure I understood, but
I probably found it somewhere in the presentation. Like,
that if you have more transcription, then you have less polyadenylation.
less
Or sorry?
deadenylation.
less deannulation. Why?
Because I wrote it in Hebrew that like more RNA staplers attached to it, but I don't really know what that means.
Okay. Okay. So maybe we can look at it later if you want and see what you're referring to here. I didn't write down, but the correct answer here is B. Okay. So next, we're gonna go over the article, which I hope. First of all, the articles I need to state this is like there will be questions in the exam from the articles. Okay, like I said, also in the beginning, all the questions which will be in the exam. I don't intend to them to be complicated. Okay. You just have to generally know. You don't have to, even for most of them, you don't have to understand. You just have to know. Okay? Like what the article is talking about. In general, a lot of time, it's really you can guess like the question that I could ask from the question that we're asking about the specific article. Okay? So like what are the components that are described in the article, what do they do, what did the researchers discover about these components, okay? So, yeah. I didn't? Yeah. And also I put the presentation for today for the topic of today, and this a few minutes ago. So if you're with laptops, then you can see the presentation, not of the article, but of the. Yeah, normally I think I didn't put these in advance because I didn't want you to I wanted you to tackle the article by yourself, especially this article which I didn't put any guiding questions in general. But okay, so. This article talks about regulation of synaptic structure and function by FMRP-associated microRNAs, 125B and 132. So, what's FMRP?
I don't remember what it's exactly.
Okay.
But it's a protein in which, like in people that have Fragile X syndrome, don't have transcription on it because of a wrong motor region. And this protein is associated with tons of stuff. Like this is only about one of these functions. Something that regulates synaptic and dendritic fine structure. And they show how through the mRNAs it regulates that.
Right. Okay. So a little bit background about this protein in general is that it's encoded, and that's important. It's encoded from the FMR1 gene, and in general, there's large variability in these repeats of the CGG, the trinucleotide repeats. They're present in the area of the 5' untranslated region of this gene. Okay, so this is like the coding region of this gene, and this is the areas of the 5' UTR, and this is the 3' UTR. And what happens is that there are different effects if you have different numbers of these CGG repeats. Okay, so if you have like less than 55 repeats, you're considered not in risk to develop like the fragile X, the mental retardation syndrome. If you have between 55 and 200 repeats, then you don't develop it during your life, but you can carry the gene. Yeah. You can carry the gene that has like 55 to 200 repeats, and then you can be like at risk of some kind of random development that can happen that can lead to having like more than 200 repeats, which is what is described in the article, which actually causes modification. We're not going to talk exactly what type of modification to this region that actually halts the translation or the transcription, sorry, of this gene. Okay, so if you have more than 200 repeats, then you don't have the transcription, and this protein, the FMRP protein, has many functions. And in this article, they talk about one of the function of this protein. So the players in the article here are FMR1 gene, like I showed you before, the FMRP, or the fragile mental retardation binding protein, that has RNA binding domains, AGO1 that also has binding domain that mediates, that is known to be part of this RNA-induced silencing complex that we talked about, the RISC complex, and microRNA 132 and microRNA 125b. Okay, so a lot of time when you read an article, if you look at it, if you try to decompose it to the different elements that they're talking about in the article, then you can try to sketch for yourself like some kind of a diagram, like you see here, about the relationship between different components and exactly what the researchers are trying to prove, and what they proved and what they didn't prove. So what is already known before they started the article, and you can learn this from the introduction normally, that this mutation, or this addition of the 200 3 nucleotide repeats in the FMR1 gene halts the transcription of the FMRP gene, and that the FMRP gene has some kind of association between microRNAs in general. Another thing that they know is that AGO1 has some kind of association to FMRP, and that microRNA 132 is one of the regulatory elements that is important in dendrite development. Okay, that's like the stage. That's what's known in the literature before. And when I mean like associated, it means that they don't know exactly what kind of interaction, like we saw in the crystallography, that they like knew the specific domains that bind some element to another. Here it's not the case. They just see like some kind of association, and in a second you'll see what I mean by association. So, yeah.
When in the pattern environment, you said that we have to describe what's known in the field before the article.
Yeah.
It's supposed to be given in the article.
Yeah. Normally it's in the article, but if you have like some kind of missing, so for example, I had to read this article, I had to look at it, to find a little bit more information about the FMR1 gene and exactly what happens there, like for myself, So that's why I looked at and I added this slide, which is not from the article, it's from Nature, like.
Just explaining why this gene is interesting, but it's not really necessary to be the article.
Yeah, but it's important for you to understand and to put the article in context and to see the importance of the article also. Okay, so what is unknown, or what are they trying to answer here, is that is the association between the FMRP in the microRNA a direct association, or maybe it's mediated by a different type of secondary association. And what I didn't add is that this association is known. Okay, AGO1 is known to. This is the operational mechanism of AGO1. It binds to microRNA. There is also large crystallography data that shows binding of AGO1 and AGO2 to microRNA. So this is known. The fact that, but the association between FMRP and AGO and the microRNA is not. It's not known. And also, when we say the association between FMRP and the microRNA, we mean who are these microRNAs and what do they do? Okay, so these are the main, and these are actually the main questions that they answer in the article. Here, you get the feeling that they're going to answer it, and actually they have the ability to answer it. I don't know why they're not answering it. Maybe it's like a political thing, but they mainly address these two questions. Okay. So, I don't know. There are certain things that you don't know a lot of time about articles, that you see some kind of like in the discussion period, we're going to talk about it in the end, they say that they still can differentiate between these two states, but they show some kind of evidence that point towards this association. Okay, but in the experiments that I'll show you, there can actually, I'll show you why exactly I think that they could have answered exactly this question, if it's associated or not. And so what they did in this experiment is that they want to see which type of microRNA are associated with the FMRP protein. So what they did is that they took a specific antibody, okay, against FMRP protein. And how did they show that it's specific? They showed that in animals that are knockout of this gene, meaning that they don't have this gene, and then they don't see, and this analysis is Western blot. Again, you're supposed to by now identify, like they call it immunoblot. It's another name for Western blot, but in general, what you see here is the binding of this antibody, and it's supposed to be in the right size also. They don't show the size marker, but here the results are pretty striking. Okay, so it's obvious that here you don't see anything, and here you see the protein. Okay.
So I don't understand how they still recover some miRNA from the knockdown.
Yeah. So you're right that this is one of the most problematic things that I saw in this article. Is that? But let's first describe like what they did, and then we'll talk about that because it's an important point. Okay. So they say they don't know. Let's say inside the cell, like I told you many times, there are all these complex interactions, and we don't exactly know like which protein is associated with which protein and which protein is bound to which protein. But we want to see. They wanted to ask, are there microRNAs that are associated? And that's why I mean associated, because they don't know. So you can have multiple proteins. Somewhere along the line you can have the FMRP protein, and then you can have a microRNA that's bound to here, or a microRNA that's bound to here, or some kind of association. But so you don't know exactly the association, but you want to see which one of these microRNAs is associated. So what they did is they actually they did a process called immunoprecipitation, which means that they used an immuno because they use antibodies, and these antibodies are normally attached to like specific elements or beads or some kind of heavy elements or magnetic elements that you can later precipitate or pull down, either by centrifusion or magnetic force. And you actually isolate this fraction from the rest of the cell, okay? From the rest of the components that are in the cell, and then you can just through types of chemical purifications, you can isolate only RNA molecules from this complex, and then with a process that is I don't think they were going to learn about it, but I think you're going to learn about it with Ami and Inbal, which is called qPCR or quantitative PCR. They can know the relative amount of the microRNA compared to something. Okay, so a lot of time you compare it to the experiment, to the control group, or you can compare it to some kind of other like RNA or other gene or something that is inside your sample. And what they saw here is that with this method they saw a high enrichment, meaning that relative to what they, and this is now the question of how exactly they did the experiment and what they compared to. They actually compared it to a state where you have a mouse that doesn't have this protein at all, and they still use the same antibodies, the same beads, and all the experimental system was the same, and they compared it to like the noise or the background level of the microRNAs that you will find just out of the technical imperfections of the process. Okay, so because no biological separation or purification is 100%. Okay, so you always have a background level. You always have some kind of contaminate level or some kind of impurified fraction. Okay, so for example, together, when they did the immunoprecipitation with the protein, then they also had a non-specific association of certain elements that can associate to the beads, they can associate to the antibody, and these are like the background, okay? The background of the experiment. So what they actually did here is that they compared it, the comparison here is between the knockout mice, which actually, because they don't have the protein, it's actually just the background of the immunoprecipitation, to the levels of the microRNA that they saw enriched after the immunoprecipitation. Okay, so it's kind of a problematic comparison. A lot of time, it's not an ideal comparison, but still, these experiments are a little bit complex. So this may be one of the.
So why do they have to measure in knockout mice? Why not just measure, do PCR to the whole block is what they want to measure is the background noise?
Well, they want to see if they took, one of the things that they said here is that when they compared the level of the microRNAs in the knockout mice, in the total brain of the, like if you take the whole samples and you don't do immunoprecipitation, if you compare the levels of the microRNA from the knockout and the wild type, there are no differences in these microRNAs. Okay?
Yeah,
So,
that's like the first thing we don't understand. So what are they measuring here? What did they take out when they did the immunoprecipitation to the FMRP antibody in knockout mice?
so what they're measuring is these elements. These elements that associate or result from the imperfect purification process. Okay, imperfect or not perfect precipitation process or enrichment process.
These elements are relevant. We don't even know if they're related to FMRP in any way.
No, they're not related to FMRP in any way. But the thing is that you want to see. In the end, the question that they're asking here is correct. They want to see if there is association or not. So if you would see that there is no difference, like in these genes over here or in this microRNAs over here, then these just arise from the background. Okay. So if you get the same level of this microRNA in both conditions, it means that it's not associated to the FMRP. Because the level is
Specific kinds of background. They just decided that, like, if I understand correctly, then you put the antibody in. If there is FMRP, it will take mostly FMRP and the stuff that's connected to it,
Yeah.
and some other stuff.
Yeah.
And they put this antibody into the knockout mice and took out only the other stuff.
Yeah.
So what mRNAs are attached to it, but it's just like we decided to pick sort of randomly this part of the background and compare it to it.
Oh, it's the only part of the background that this is like the ideal background that you can test because it's like because it has everything but the FMR. Okay, so any unspecific association that happens in this protocol, they will control for in a sense. Okay, if you have association.
Do the immunoblocking first also. They take out like or they skip that phase. They take out like only stuff that's attached to the FMLP antibody. So everything they do after this is already not the full background because it's only part of the protein.
Yeah, but still there are procedures. You have solutions that are passing through the actual procedure that are in charge of this background. Okay, I understand like where you're coming from, but in a sense, you need to ask like, what will happen if I have a microRNA that's associated to FMRP? Will I see this profile if I do this experiment? And it's true that if you have a microRNA that's associated to FMRP, then you will see this pattern. Okay, this pattern of enrichment. And if you have a microRNA that's not associated, then you'll see this pattern. Because it will just be the background. Because you pull down, it doesn't matter that you pull down FMRP, it doesn't have any microRNA, it doesn't have this microRNA attached to it, so the only level of the microRNA that you're going to see will be the background.
So why do the immunoblocking
This?
Like,
This?
For the knockout, like for the wild type, I understand they want to see specifically what's attached to the FMRP.
Yeah.
But I don't understand why they're comparing it to something that they did through immunoglobulin.
Because it's the ideal background. You do the same steps of the whole procedure, just that you don't have the protein itself. Okay, so that's the only difference. So it's just like the background level. So you do everything. The way they did the experiment is exactly the same for all of these samples, and they extracted everything from the same benzoyl gel, and they took everything from the same location. So they have a better representation of the actual background, or the actual microRNAs are just randomly.
If they compare to the actual background, it's the same.
No, I didn't say the background. If they compared it to RNA that is extracted, that RNA that is not immunoprecipitated, meaning that it's taken from the full brain, then you're going to see that there is the same levels of these microRNAs.
Okay, but if you compare the, like in the FMLC mice, like the wild type mice, you do the immunoblogging and you compare that to the full background. So just like how many of these mRNAs are really out there.
You can also do that. You can also compare just inside the wild type, like you said, like take the immunoprecipitation and compare it to the ranking, to the order of the microRNAs inside the actual brain. It's also a legitimate experiment that you can do, but there you're not controlling for the background. If you have some kind of microRNA that is has very strong association with the beads, then you will still see it enriched, but it will be false. Okay, so again we can. Okay.
Also, I didn't really understand why they took multi-specific mRNAs. Some of them they said that they're known to be related to blood and stuff like that, but then they just go, like most of them, they just say like
they're not.
Average and low abundance.
Yeah. So the thing is about most of these microRNAs, they're famous microRNAs. Okay, they have research that's related to them, and they also know the relative abundancy of these microRNAs. For example, 124 is very abundant in general in the brain, and 132 is not very abundant. Okay, and this is like medium abundancy. So you can see also that they didn't get like enrichment of microRNA that have higher basal levels. For example, this can be one of the controls, but. You're right, again, in your instinct, that why did they ask specifically this? Especially this experiment was done in 2010, and today we have much better techniques than qPCR to ask these questions, and we can get all the microRNAs, okay? So why not ask? They're not a lab that has like money issues or stuff like that, so that's one of the questions, why didn't they do like all the microRNAs? I don't know, but one of the reasons may be that they have another article. They want to take out another article from this data, so they just publish part of the data, which is illegal in a way. But yeah, there's a lot of questions behind it. Like I said, in like the politics. Okay, so let's just go very fast over what they actually found. Is that here it's important to understand the experimental. Like, how do you say? The experimental motif, so the experimental design, I mean, is that they looked at primary neuronal culture, meaning that neurons that were taken from mouse embryos, they were seated on the plate, they were led to like freely differentiate into mature neurons and develop synapses and dendrites, and then they looked at the morphology. And what they did in these experiments is that they overexpressed these microRNAs, okay? So they overexpressed microRNA 125B, and they overexpressed microRNA 132. And what they actually saw is that it has, and this is the main actually result of this article in general, they saw that it had dramatic effect on the shape of the actual spines. Okay, so this is very, very drastic. Okay, the effect that you overexpresses microRNA 125B, and it has this effect on the spines, and 132. So 132 they say that the spines are more stubby and short, and 125B are more long and thin. So they actually have like this reverse effect of overexpressing these two microRNAs. And then the next question that they ask is that this experiment doesn't have anything to do with FMRP. Okay, they showed the experiment before that showed that there's an association between 125B and 132 and FMRP. And also they showed in a separate experiment that if you overexpress these microRNAs, you see these effects. But are these effects mediated by FMRP? So here they say that what they did is actually they used the shRNA, and actually this is a fancy word to say it's like siRNA in a sense. It's a way to introduce silencing of this FMRP mRNA, or degradation of the mRNA of the FMRP, and thus leading to lessen the amount of this protein. So that's what they actually did here, and and again they tested morphological features for these spines. So I don't want to talk too much about this figure because it's complex, because you see that there's these complex interactions, but in general, in some of the aspects, you can see that if this is and this is their background, okay, so in general, you can see that the effect that you have, the length of protrusion for mere 125B, is diminished when you knock down the FMRP1 protein, okay? So there, you look puzzled? Yeah, okay. Oh, in all these FMRP number 1 is just the type of vector that they used in order to introduce the siRNA to knock down the gene, okay, or to knock down the protein. And I'm not saying knock out because they didn't genetically modify it or something like that. But when you have lower amounts, so in these conditions over here, you have a lower amount of the FMRP protein, okay?
They said that the FMRP alone doesn't do these things, and
Again, what?
like if I understood the article, they said that FMRP alone doesn't do any effect. It needs the miRNA. But if you compare the 143, which is like the NONA experiment,
Yeah.
In the third condition, there is a significant difference just when they have FMRP and don't have FMRP.
Yeah.
So it's not the effort. Sometimes FMRP makes a difference.
Yeah, it has, in this case, the FMRP has itself, the knocking down the FMRP itself has an effect on the protrusion density if you also if you also overexpress mere 143. Okay, so yeah, you're right in that sense. So. And again, I didn't want to get into the interactions here because they have all kinds of weird things that you can understand from here and the interaction between these two. But let's focus on this. And in general, they see that this effect of mere 125B is abolished by the changing of the levels of the FMRP1. Okay? Question? So again, in the question of what do they do? What do these microRNA do? And they use a really nice construct, and in order to overexpress the microRNA, you introduce some kind of circular DNA. We'll talk about it extensively in the next class. But you can cause this overexpression of the microRNAs, and in order to reduce the endogenous levels of the microRNAs, they introduce the construct that is called, today people often use it as a sponge, okay, and also here they call it a sponge, is that this special type of RNA molecule that has multiple sites that actually have very good complementary with the microRNA, except for a specific region that is critical for the cleavage of AGO2 in this case, okay? So, or the cleavage of so if we would have perfect complementary in this sequence, what we will get is the cleavage of this sponge, and the effect would be lost very fast. Okay? But what we actually want for this sponge is for it to absorb the microRNAs, for the microRNAs to remain bound to it, and then just nothing would happen to this mRNA.
So if it's still expressed,
The microRNA is expressed, but it's busy to it's drawn to the special mRNA or the special RNA that actually that has a very strong affinity to binding this microRNA. Okay, because the complementary is very, very good, except for this small loop that is here to because this is actually like the site of complementary that the AGO2 needs to cleave the mRNA. So they actually manipulated that, thus lowering the amount of the endogenous, actually the actual available amount of the microRNA. Okay? And again, what they see here is a very nice reverse effect. And the way that, what do we actually see here? What did they actually measure here? Do you know? This is important as neuroscientists. Okay, so these are currents, yeah, but what? Of what? So before we talked about synapses, okay? One of the measures to determine like synaptic strength in general is the proportion or the amount of what we call miniature release. Okay, did you discuss minis in general? Do you know this concept in general? Yeah. So minis, and I think that with this figure we're gonna finish. So minis or miniature release are actually vesicles that are spontaneously released from the synaptic cleft, just like as a basal level. Okay. So like everything in biology, nothing is obsolete, like I told you before. And if you're looking at the synaptic cleft, by now I'm supposed to be able to draw synapse very fast, but still. Okay. So we have these vesicles, right, that are fused to the presynaptic terminal, but actually this whole condition here, in order for it to be effective, in order for the synapse to be readily activated, everything here needs to be like balanced, or it needs to be like potentially active. Okay, this whole mechanism of the synaptic release, or the vesicle fusion and the synaptic release. So what actually happens in synapses is that you have this kind of constant flow of synapses, like a small fraction of synapses are always like fused, and a little bit of neurotransmitter is released. Okay, and these small events look, when we record from the cell, they look like these small EPSPs, excitatory postsynaptic potential in this case. But in order to see these small currents, you have to do two things. Okay? And this is just to wake you up a little bit. This is a fogo or catfish, right? It's a catfish or something like that. Anyway, this special type of fish has a very potent toxin, and this toxin, which is actually TTX, I don't know if you heard about it, but it generally blocks sodium channel or voltage-gated sodium channel and completely prevents the generation of action potentials. Okay, so this is the first thing that we need to do, to add TTX in order to block action potential, because we don't want it to mask these minis. And the second type of things that we want, in order for us to see only, like a lot of time, if you want to see like a specific signal in a specific direction, like excitatory postsynaptic potentials are not a mixture of inhibitory and excitatory postsynaptic potentials, you need to inhibit one of them. And this is what they actually did also in this experiment here, is that they added the picorotoxin, which actually is an inhibitor of the GABA, of the inhibitory channel of GABA. Okay? So they actually blocked all inhibitory currents, and they blocked all action potential, and what is left is just like synaptic inputs which can actually they cannot arrive because all the cells are not able to fire action potentials, okay? So if none of the cells can fire action potentials, then there can be no like full synaptic releases, and all that is left is these minis, or these excitatory minis, okay? So this is what you actually see here. Okay, imagine there's a red arrow here. But what they actually saw is that when you overexpress microRNA 125B, it really reduces the synaptic strength, meaning reducing the amount of minis that are released. Another important thing is that the amount of minis that are released are often a measure of the synaptic strength. Okay, if you have like
Exactly the same measure, like.
they measure from the cell body.
From the cell body of the synapse, like after of the cell absence of the postsynaptic cell?
Yeah, it's always it's always like
No,
all of the cells are
they
postsynaptic.
want to measure a certain synapse, so
They want to measure a certain current, and thus they block other currents. But you always measure from a cell, and this cell always receives inputs from somewhere. So it's always a postsynaptic and a presynaptic, but you.
According to the synapse that we want to check like the currents of a specific synapse. So do we check?
It's not specific synapse. These currents, for example, this can come from one synapse, and this can come from another synapse. Okay, the different types of currents. Okay, so it's from all excitatory synapses that are connected to the cell that they recorded from. So you can see that here you have overexpressing the mirror lowers the synaptic strength and the amount of minis, and the opposite thing happens with mirror 132. When you lower the amount of the microRNAs, you have like the same effect. So again, we see these inverse relationships, and actually in the sponge, they didn't see, in the microRNA 125B, they didn't see a large effect. It's more or less the same. But in microRNA 132, they saw exactly also an inverse effect inside, like upregulating the gene or overexpressing the microRNA caused an increase in the synaptic strength and the miniature release, and here it caused a decrease. So it's really like a more of a one-to-one relationship. Okay, so we'll have just one more slide, and we'll continue with the next class. We'll take 15 minutes break or 10 minutes. Is 10 minutes enough? Or 15 minutes? Okay. No problem. And we'll return to the rest of the article, and then we'll start the next chapter. אחד בין microRNA, כאילו לבין רגע שתראי שהmicroRNA שאנחנו עושים בעבודה
תסתכל אני אגיד לך.
כי אם כן, אז אני אעבור עכשיו לכיתה הקטנה.
אוקיי, דקה.
אוקיי.
שלום. כן. בסדר, אני קראתי מה זה. כן.
אוקיי, אנחנו נעבור לכיתה הקטנה, יש פה שיעור.
רגע, אבל יש לך חשבון ב...
אני מחשב לא מספיק?
לא, אתה צריך...
להירשם?
כן, כי אם מישהו יתפוס מקום יבוא יוצא אותך החוצה.
אתה יכול לרשום אותי חד פעמית? זה רק היום. פשוט עד 2?
אני לא חושב, זה נרשם עליי. בסדר, עד 2?
כן. זה עובד עם לחיצה הדבר הזה? תשמע, כאילו... אני לא פה היום?
לא.
גם היום לא פה? רגע, אני מכבה את המקרן או אשאיר אותו דלוק?
לא תחבר, אם אתה סוגר את הדלת זה גם נפח. טוב, מה שמך?
נדב?
נדב.
עושה מולקולר נורוסייאנס.